Today I'll be talking about the adapter hypothesis that was proposed by Francis Crick in the mid-1950s. The adapter hypothesis is in a sense a part of the central dogma of molecular biology. The central dogma consists of replication, transcription, and translation. DNA replication occurs and then the DNA is transcribed into RNA. Since both DNA and RNA are built with nucleotides, there is no essential language change between the two. However, going from RNA to proteins, there is a change in language, making it a translation from nucleic acid to amino acids. So what allows this translation to occur? How does the body just translate between two languages of monomers so quickly? With the use of ribosomes and an adapter protein, as called by Crick, the nucleic acid can be rewritten into amino acids to produce proteins. mRNA is the ending result of the transcription process of the central dogma, and it carries the genetic code. Ribosomes then can read the mRNA and attract the corresponding adapter protein to help translate the genetic code. The adapter protein was later classified as tRNA through additional studies. The ribosomes attract the corresponding tRNA to the mRNA. tRNA brings amino acids to the ribosome and helps match the correct amino acids to the mRNA that holds the genetic information. The chain of polypeptides created by the tRNA becomes a chain of proteins. The process is shown in the following slides. The mRNA sequence reads as AUG, AAG, CCA, AGC, UAA. Ribosomes then attach to the mRNA, which allows the tRNA to attach. The appropriate tRNA molecule attaches to each triplet. With the tRNA, the corresponding amino acids follow. The amino acid chain is then built from start to stop. Why the name adapter for both this hypothesis and the protein? Crick knew that there was an adapter involved because there was a translation in language. There had to be a molecule that could understand both amino acids and nucleic acid. tRNA can do both and code for the specific amino acid needed. tRNA can act like an adapter to connect two different languages correctly. The genetic code is read in triplets, meaning 64 base code pairings are possible. However, there are only 20 amino acids. This means that a single amino acid could code for multiple base triplets. tRNA follows a universal genetic code in which RNA is paired specifically with a specific amino acid. What if an alteration occurs that prevents tRNA from acting as an adapter that follows the universal genetic code? Without tRNA, the body would have to find another method to translate between the two languages. Mutations would occur within the protein chain and cause the body to react badly. The wrong amino acids would be coded for, and the body would not have the proper methods of start and stop, since there are specific amino acids that address the starting and stopping of amino acid chains. tRNA is a very important molecule for the body, and as an adapter protein is one of the only means of communication between the genetic code and protein chain created. These are my references, and thank you for watching.